66th of the Beyond Research Where Curiosity Meets Conversation webinar series, which is co-organized by Biopractify, the ACS student chapter of IIT Delhi, and Biolabs, a research division of Biopractify, where our speaker, Miss Ayushi Singh, is joining us today, where she'll be sharing insights about from bacteria to parasites, how molecular insights into resistance and novel therapeutic targets. And welcome, Miss Ayushi, for the session today. Before we start, I'll just like to give a brief introduction about our speaker today. So, Miss Ayushi is a researcher at the CSIR CDRI, Lucknow Division of Molecular Microbiology and Immunology, with a strong foundation in infectious disease biology. Ayushi has an outstanding academic journey, clearing IIT JAM GAT B with an AIR 7, CSIR NET JRF, and GATE Excel and BT. And after completing her master's, uh, I think the voice is echoing. Yeah, now is this okay? Yeah. So after completing her master's in science and biotechnology from IIT Roorkee, her research spans from structural and functional characterization of bacterial enzymes involved in antimicrobial resistance, earning her a cover page co-authorship in the ACS Biochemistry. To advance malaria research, currently pursuing her PhD at CSIR CDRI, she works on genetic knockouts in Plasmodium burgei to uncover resistance mechanisms and validate novel therapeutic targets, making today's session a unique bridge from bacteria to parasites and from molecular insights to translational impact. So, welcome, Miss Ayushi Singh, for the session today. We are really eager and delighted to have you with us as a speaker for the session and looking forward to your insights. Over to you.
Thank you so much, sir. Am I audible?
Yes, you are audible.
Let me share my slides first. Mm, Are my slides visible?
Not yet.
Okay, wait. Now, sir?
Yeah, they are still loading, I think. Yeah, they are visible, but
Okay,
not
okay.
in slideshow.
Yeah, yeah. Then
Okay.
I think it's fine now.
I think you'll have to share your entire screen. Maybe then it would be visible clearly.
Sir, I'm sharing my screen. I think it must
Okay,
be a
so
network issue.
I think it's a network issue on my end. Are the participants able to see it? Can anyone confirm?
No, sir. Right now it's shared.
Okay, it is visible now. Yes.
It is visible?
Yes.
Okay. So, shall I start?
Yes, you can start.
Okay. Thank you so much, sir, for the introduction. So, I'm Ayushi Singh. I'm currently pursuing PhD from CSI CDRI, and as sir introduced me, I did my master's from IIT Roorkee. So the work I'm presenting today, it's based on my own work which I have done in IIT Roorkee, and the same theme I have continued in my PhD, which is microbes. So as the name of the program is Beyond Research, Curiosity Meets Conversation. So talking about curiosity, the word microbe sparked curiosity in me in my bachelor's. I didn't have any background of biotechnology or microbiology, but the term microbe itself, that they were the first living organism on this planet when there were no suitable condition of living. They still thrive in the harsh climates of this planet where no other organism can live. That thought that they have such an excellent survival strategy that sparked curiosity in me, to study them. So I always had an inclination towards microbes. And a microbe is a fairly broad term, not just bacteria. It includes parasites, eukaryotes, prokaryotes. But in my master's, I got to work in an AMR lab, which worked on beta lactamases and protein biology lab. So there I started my journey in working with microbes. So in this whole theme, I just wanted to convey that what is antimicrobial resistance, how it is affecting us today, why it should be stopped and how it should be stopped. That whether it is a bacteria or a parasite, understanding the molecular machinery behind both is the key to novel therapeutic targets. That's what I want to convey. So starting with why this topic actually matters. What is the importance of AMR? So, like, if many of you would just go through your social media or LinkedIn, many of you will hear the term that WHO said that it is a global public health and development threat. Why so? So, AMR is causing more than 1 million deaths globally and annually, and it is increasing by a rate of 15 to 20%. Like, isn't that scary that it is not a disease? These are just small infections which can be cured by a drug, but they are become so resistant that not even the last resort drugs can work for them. So what happens is that there are direct deaths, 1 million direct deaths due to AMR globally. Apart from that, there are many other cases in which AMR plays role, like for example, in a hospital setting when you're having a transplant. So in that situation, your immune system is very weak. So you need to be protected from these infections by certain antibiotics. And these infections, they are caused by opportunistic bacteria. They are not very harmful. But these opportunistic pathogens, they have evolved so brilliantly that these first line, second line, third line drugs, they are of no use now. So what happens is that you can come out safely of a surgery, but just a microbe, it's that all it takes now for a death, for a casualty now. So that serious it is today. That's why everyone should be concerned about it. Like you guys must have heard that whether it be a viral infection or any kind of infection, doctors nowadays they are very used to giving antibiotics. Even people don't know much about it, but they just take for a sore throat, they take azithromycin for a day and they just leave it because they feel better. So these are the causes which are causing this antimicrobial resistance globally. This this not taking dosage properly, not under guidance of doctor, indiscriminate drug use, these are all causing antimicrobial resistance to rise. So because there is so much resistance, there is a constant need for new targets, new novel targets which are foolproof. Therefore, to understand it, on let me take you to the why this resistance happen. So I'll be using Hindi also sometimes so that some of you can understand. तो क्या होता है कि there is a population of bacteria, okay, one or two got mutated, casually got mutated because there is so much frequency of replication. There are one or two mutations which are not very important at that time, but just imagine there is a drug pressure, indiscriminate drug use, and that mutation proved to be helpful for survival of that bacteria. So what happens that during the course of time, those bacteria get selected because they are fit for survival and then they get replicated. तो क्या होता है कि during the course of time we get the population of these resistant bacteria. That's why if you see in our day-to-day life, not making it too scientific, but in our day-to-day life, when you use Mortine or these coils for mosquitoes, on day one or day two, you will see that there will be less mosquitoes. But if you'll keep using them every day, you will see in a week or 2 weeks, mosquitoes will still be the same. It will even increase. because you are selecting the resistant mosquitoes by that drug or by that fumes. So this is what the example of resistance in our day-to-day lives. So why I am focusing on AMR, I'll tell you, because my scientific journey started from a lab in IIT Roorkee, a protein biology lab. There I worked on a bacterial enzyme, basically a very unknown bacteria and unknown enzyme. So I characterized it by biophysical and biochemical characterization methods, and I got to know that it had some activities which can cause resistance to drugs. It was totally unknown. It is an opportunistic bacteria, not too harmful. But why it is important to know what kind of activity enzyme has? Like if I got to know कि उस एंजाइम में बीटा लैक्टमेज एक्टिविटी भी मुझे मिली जो कहीं पहले कहीं भी नोटेड डाउन नहीं थी. Even there was a paper in protein science which said that that enzyme didn't have beta lactamase activity. But in my study, I got that it does have promiscuous beta lactamase activity. So, what is a promiscuous beta lactamase? Ki secondary activity. So why it was important to know that if this opportunistic bacteria, we are treating it in a medical setting and we don't know that what kind of drugs it are resistant to, then unknowingly we are feeding it with those drugs and we are creating more resistance and toxicity in that patient's body as well. So that was the protein aspect of AMR that I learned in IIT Roorkee. So I worked on bacterial enzyme. After that, I wanted to move toward a more complex microbe. So at CDRI, I took part in a malarial biology lab. So malaria is caused by Plasmodium. Everybody knows that. We are reading that from NCRT days. But that Plasmodium, it is so complex, it has so many life stages, it affects so many tissues that it is even more notorious than any other bacteria. Because आप इसके कितने pathway या कितने stage आप इसके target कर लोगे. If you will target one stage, it will survive others. It's that notorious. And also malaria being a problem of developing countries, including our India, which is neglected by top giant pharma companies and developed countries, because these treating these malaria and these tropically neglected diseases, basically it won't pay them well. So that also attracted me, something that is related a problem to our nation or the developing countries. So I started my journey in Plasmodium biology. But first, I would take you through my journey of how I started the study in microbes from IIT Roorkee, which is on the enzyme from an opportunistic pathogen. Me, sir.
Sure, sure. We'll let you know.
Okay. So I told you about my scientific journey. So without taking any time, I will start with my work at IIT Roorkee's lab. So this Burkholderia gladii, many of you must have not even heard about this, but this is an opportunistic pathogen. So what is an opportunistic pathogen? A pathogen which is not normally harmful, but it might be harmful if you are immunocompromised, which is in hospital settings. Like if you're having a surgery, if you're having an implant, if you are immunocompromised, then it can cause you harm. So this bacteria, it was found very often in the cystic fibrosis patients, in the respiratory tract of cystic fibrosis patient. So we took one random enzyme from this bacteria, not random actually. The enzyme's name is STB and it already has in paper in protein science, but I'll tell you further what we did extra. So this pathogen, as you can see, the paper cuttings I have mentioned that it is quite harmful, or it does have an effect in the transplant patients. So this enzyme EastB, it is usually an esterase. It is mentioned that it has an esterase activity, but it is also mentioned it has overall fold of a beta lactamase. So what is a beta lactamase fold is, if you can guys clearly see, you can see there are alpha helix and in between there are beta sheets. So this is an overall beta lactamase fold. So this STB enzyme had overall similarity to beta lactamase structure. But in all the papers existing, they mention that it has no beta lactamase activity. But we had a curiosity that why so? We aligned it with all the beta lactamase groups as the class D, class A, and class C. As you can see, there are so much structure alignment, they almost overlap, and the most alignment is with the class C beta lactamase. As you can see, the RMSD value, which is in short the deviation value from the structure, that how much deviated it is from the class C structure, is the least in alignment with class C, which shows that it was very similar to the class C beta lactamase. Here, my curiosity spiked that if it is so similar to a beta lactamase, we even dogged it with a beta lactam substrate, which is a nitrocephin. It is often used in the assays we do in the beta lactamase test, because it is a colorimetric substrate. So, whenever there is a beta lactamase enzyme, if you mix it with nitrocephin, it will turn immediately red, deep red. That is the test, nitrocephin test we do for beta lactamases. So if you guys can clearly see, the first dot you can see in the video is the nitrocephin itself. Second is nitrocephin mixed with STB, which is my enzyme, and second is nitrocephin mixed with SME1, which is a highly potent beta lactamase. So you can see that there is not much difference between the second and the third dot, which clearly says that SB does have measurable beta lactamase activity. So the claims were wrong. So because of this curiosity, we moved ahead and did some kinetic study. So in the nitrocephin test, you can say that the enzyme has a beta lactamase activity. But why do we do kinetics? Is you get kinetic parameters. These parameter defines that how good an enzyme is, like how good or how efficient it is as a beta lactamase. For that, we determine the kinetics with different beta lactam substrate. So these are beta lactam drugs: nitrocephin, cephalothin, ampicillin, carbanicillin, piperacillin. So ampicillin, carbanicillin, piperacillin, these are a class of penicillin only. And the nitrocephin and cephalothin is first generation cephalosporin. So to make it easy, penicillins were the first class of beta lactam group discovered. Then due to the resistance, there were more changes made to it. So these first generation cephalosporins are most complex than penicillin. So in the to make this data easier to understand, let me explain that Kcat, if you can see the last row, Kcat is actually measure of the enzyme efficiency. And if you can see that the enzyme efficiency is more for the carbanicillin, ampicillin, means the penicillin, it is more. But as the complexity of the drug increases, like in cephalothin, it will decrease. Same I have shown with the bar graph, it is easier to understand that the enzyme of this activity of this enzyme is decreasing with the complexity of the drug, and it is very similar to the class C beta lactamase, because class C beta lactamase also don't cleave the higher second line drugs, which is the second generation or third generation cephalosporin. Same case we got in this. So according to the parameter, it is not very potent a beta lactamase, but it does have measurable parameter to say that it was a promiscuous enzyme. So a promiscuous beta lactamase means that primarily it is an esterase, but it does have a secondary activity which is lower. It is a beta lactamase activity. But the problem is that these promiscuous activity are actually the home for resistance. What happens is that whenever there is a drug pressure, these activity might contribute to resistance, because I don't know if you have heard of 10 beta lactamase. It is a very famous class A beta lactamase. It originally evolved from ancient hydrolases. So the ancient hydrolases in microbes, they were there to degrade toxins and just for the normal cell physiology. But because of the drug overuse, through the hydrolases they evolved. So it was a promiscuous activity which became a resistance mechanism. That's why studying the promiscuous activity is very important. Even we did the inhibition kinetics as well to see if how much similar it is to beta lactamase. Does it bind its inhibitor as well? So it is a very famous inhibitor of beta lactamase is avibactam. It is the most recent inhibitor. It is given alongside with the beta lactam drug to increase its efficiency as a drug. So we found that it binds very strongly to avibactam as any beta lactamase would. As you can see in the graph, that increasing concentration of inhibitor, the enzyme activity is decreasing. See the lines, they are going upside, and eventually with the increasing inhibitor concentration, the activity is decreasing, means the increase of binding strength. So that is very consistent with the data that it is a promising beta lactamase activity. So what the key lesson I saw from this study, that the resistance is a very serious problem today. It's not a problem of the future, it is a problem today. Molecular studies are important to predict these pathways and to search for novel therapeutic targets, because no matter how many drugs we are making, the speed of evolution of these microbes, they are faster than the drug pipeline of our pharma companies. So it is very important that there is a constant molecular studies to search for novel targets. Also, knowing resistance help us to give the proper treatment, to administer the right treatment, because if we are not knowing that what kind of drug this particular microbe is resistant to, we are increasing the risk to the patient by increasing the toxicity of the drug, and indirectly we are contributing to more antibiotic resistance. Therefore, molecular studies are the basis to stop or to even decrease AMR globally. So here comes my transition from bacteria to parasite. This was all about bacteria. After that, I wanted to have experience with some more complex microbe. So I chose malaria particularly because it intrigued me that it is such a big problem in developing countries like India, Southeast Asia, African countries, but it doesn't bother globally to invest in R&D for malaria. No giant pharma companies are invested. It won't give them huge paychecks. That's why there is very less budget, R&D budget for malaria. But this parasite is so smart that if you will target its one stage, its other stage will still be there. So this is the overview of a plasmodium. Plasmodium is the organism which causes malaria. There are many species of it, particularly falciparum is the deadliest in humans. Vivex is the most widespread, which we hear often. The malaria which we know is the Vivex. And then there is Knowlesi, which comes from monkey. And then there are Burgii, which is a rodent malaria. These are the well-known species. So this is a common stages of a typical Plasmodium parasite. So as you can see here, it starts from the mosquito bite. This mosquito is already infected with Plasmodium sporozoites. These sporozoid stage, they are motile. When the mosquito bite, these motile stages through the bloodstream, they reach your liver, hepatocytes, and in hepatocytes, they multiply. They multiply and change to a different stage called merozoites. And these merozoites eventually form schizons. Schizons is the stage where the wall of this parasite is most permissible. So when we make these genetic knockouts, which I will tell you further, in genetic knockouts, what we do, we make a particular gene non-functional in Plasmodium to study what effect it will have on the parasite. So for that to happen, you need to transfect the parasite with our made construct of the genetic construct which we want to replace that gene with. So for that transfection purpose, we chose schizont because its wall is the most permissible for DNA uptake. Okay, so these all stages, merozoid, schizont, these are blood stages of parasite. It is taking place in human host of Plasmodium. Then these schizons, some of these schizons form gametocytes. Gametocytes, when they ingested by the mosquito, it goes in the mosquito. It is not mentioned here, but I'm telling you verbally. Gametocyte, when ingested by mosquito in a blood meal, they go in its gut. In the gut, it form gamete. Gamete will fuse to form zygote. Then that zygote will form a ookinete stage, which will penetrate the gut wall and form oocyst. That oocyst will grow and grow, and eventually inside that there are many small sporozoites forming, and as it will grow it will rupture, and these sporozoites again will migrate from mosquito's gut to its salivary gland, and again the same cycle start. Sporozoid to liver, liver to blood stages, blood stage to sexual stage, and back to mosquito. So as you can see, it is affecting so many tissues and it has so many stages that what you can target. That is the challenge we face in studying malarial biology. This parasite is way smarter than I guess all the humans combined. That's why we say microbes are smarter than human, because otherwise there are cure for so many big diseases, but still now we can't have a cure for a common cold. That's the power a microbe holds, I guess. So Plasmodium, as I told you, it has a complex life cycle with stage specific biology. So it has genes which are expressed at certain stages and not expressed in other stages. So there is a very tight gene regulation in parasites, more important than in any other organism. And there are very limited number of drugs, not like beta lactam drugs. There are many variety, but for malaria there are not much drugs. Like in the starting in 1940s there was chloroquine, after that there were antifolates, and right now there is artemisinin combination therapy, which is the latest and the foolproof treatment they say. But there are news that there are artemisinin resistant strains also evolving, even in India as well, due to years of drug pressure. So it is important to study Plasmodium as well. It was once among the big three on the WHO's list, who had WHO had these three big diseases they call big three, which had HIV, hepatitis, and malaria. Artemisinin resistance. If you can see this paper cutting, particularly this one, I am a student of CDRI. I live in Lucknow, and this paper cutting I saw in one of the newspaper. It is very recent in September, that a nearby town Sitapur there were many cases, spike in malarial cases. So there were very immediate treatment in the shelters. That was the case. And in year by year, these cases are increasing. But because not globally mass people are affected, that's why many people don't care about this. but it is a problem of our country as well and other developing country as well. So we need to look into it, the Plasmodium molecular machinery, to find our solutions. But the resistance in Plasmodium actually is little different from bacteria. So in bacteria, what was happening, that bacteria produced enzyme that cleaved the drug. It destroyed the drug. But in parasite is so smart, it don't need to destroy the drug as well. It it is causing resistance without direct drug degradation. So how does parasite do that? How Plasmodium does that? It has stress responses. It hides from the drug. What happens is artemisinin, actually what it does, it is activated by the heme of RBC. The Plasmodium, when infects RBC, it digests it, so the heme is released. So because of that heme component, artemisinin is activated to destroy Plasmodium to its cell wall and cause pores in its cell wall. But what happened is that parasite, it cause less heme destruction to escape from artemisinin, due to which there are artemisinin, partial artemisinin resistance evolving even in India. Therefore, we need more novel targets for Plasmodium therapy. So, why am I studying Plasmodium genes? Why do we need to study Plasmodium genes? Is because 40% of the Plasmodium genes are uncharacterized. We make knockouts of Plasmodium genes in our lab. We have been making it from 15, 16 years in our lab, and many people are doing it from so many years, even in abroad. There is a very famous malarial group in Harvard as well, but still 40 to 50% of genes are uncharacterized because they were not similar to human or any other organism. That means these genes are very parasite specific. So if these are parasite specific, we need to know about it, that what role is it playing in the parasite. Because if we get to make these as a drug target, there will be no off target effect on any patient. So that's why we need to know about Plasmodium genes. So talking about the work, my current PhD work, the overview, I'm giving you the overview of the work which we are doing in our lab. So we select a gene randomly or by reading any literature, and if we are interested in a machinery, we take a gene and we make knockouts of it. How do we make knockouts? Is example, if you can see, there is a gene X. We need to knock out or remove this gene X. So to remove this gene X, we use a simple biological process of homologous recombination. सो होमोलोगस रिकॉम्बिनेशन आप सबने पढ़ा होगा, एनसीईआरटी में भी पढ़ा होगा और अपने पाथ फाइंडर और सब बुक्स में पढ़ा होगा, दैट इट इज हमारे सेल्स में भी होता है फॉर स्ट्रैंड रिपेयर में होमोलोगस रिकॉम्बिनेशन होता है. वही सेम लॉजिक यहाँ हम यूज करते हैं कि जीन एक्स के लेफ्ट और राइट साइड का जो एरिया है, जीन्स हैं, उसके हम होमोलोगस रीजन बनाते हैं. होमोलॉगी 5 प्राइम होमोलॉगी आर एंड 3 प्राइम होमोलॉगी आर, वो हम पीसीआर से वो इंसर्ट्स बनाते हैं. फिर हम उसको एक वेक्टर में क्लोन करते हैं. उस वेक्टर में दो मार्कर्स होते हैं. एक स्क्रीनेबल मार्कर है जीएफपी, जो कि ग्रीन फ्लोरेसेंट प्रोटीन है. ये हमें फ्लोरेसेंस इमेजिंग करने के काम आएगा. और दूसरा सेलेक्टेबल मार्कर है HDHFR जीन. ये हमें नॉकआउट पैरासाइट को सेलेक्ट करने में हेल्प करेगा, क्योंकि ये पायरामेथामीन जो होता है, पायरामेथामीन इज ऑल्सो अ ड्रग विच इज यूज्ड अगेन प्लाज्मोडियम, बट दिस जीन कन्फर रेजिस्टेंस टू पायरामेथामीन. दैट मीन कि जिस भी पैरासाइट में ये कैसेट जाएगा, जीएफपी और HDHFR वाला, वो पैरासाइट पायरामेथामीन के लिए रेजिस्टेंट हो जाएगा. इट विल आउटग्रो पायरामेथामीन. So this is how we select our knockout population of parasite, because whenever we are trying to create knockout of parasite, हर हर पैरासाइट में ये होमोलोगस रिकॉम्बिनेशन हो या ना हो, ये तो एक चांस इवेंट है. तो देयर विल बी वाइल्ड पॉपुलेशन एज वेल, एंड देयर विल बी नॉकआउट पॉपुलेशन एज वेल. So to select the knockout population and create a pure population of knockout parasite, we use these GFP and DHFR genes. So what happen कि हम जब 5 प्राइम और 3 प्राइम क्लोन करते हैं उस वेक्टर में, और उसको पैरासाइट के साथ ट्रांसफेक्ट करते हैं. ट्रांसफेक्ट करने का मतलब है कि पैरासाइट की मेम्ब्रेन को परमिसिबल बनाने के लिए हम इलेक्ट्रोपोरेशन करते हैं. इलेक्ट्रोपोरेशन से ब्रीफ मोमेंट ऑफ टाइम के लिए ट्रांजिएंट पोर्स क्रिएट होते हैं प्लाज्मोडियम के मेम्ब्रेन में. तो उसी टाइम हम अपना ये जो कंस्ट्रक्ट है जिसमें हमने क्लोन किया है होमोलॉगी आर्म्स, इसको मिक्स करते हैं, एंड देन वी इंजेक्ट इट इनटू माइस. So we do in vivo study. We inject it into mice. So there will be transfection done. There will be knockout population in mice, and there will be wild type population as well. फिर हम उस माइस को ड्रग अंडर ड्रग रखते हैं पायरामेथामीन में. उस पायरामेथामीन में वाइल्ड टाइप पॉपुलेशन सारे मर जाते हैं, एंड दिस नॉकआउट पॉपुलेशन आउटग्रो पायरामेथामीन. That's how we select knockout parasite. Now that the genes, this gene X is deleted from the parasite, वी विल हम उस पैरासाइट की साइकिल चलाएंगे माइस में. क्योंकि आई एम वर्किंग ऑन प्लाज्मोडियम बर्गियाई. बर्गियाई इज अ रोडेंट मलेरिया, because it is not very easy to work on Plasmodium falciparum, because in my lab we do in vivo studies. We do the life cycle of this parasite in mice and in mosquito as well. So we establish an insectary as well. But in falciparum is so deadly that it is quite not possible to do the study on mosquito. That's why we prefer Burgii as a model to study Plasmodium. So that's how we study. सो दिस पैरासाइट का एक लाइफ साइकिल हम चलाते हैं माइस में, देन इन मस्किटोज, जस्ट लाइक ह्यूमन और मस्किटो में चलता है लाइक सेम. फिर हम उसको डिसेक्ट करते हैं मस्किटो से या लिवर से. फिर हम उसका इम्यूनो फ्लोरेसेंस करके देखते हैं कि उसका फिनोटाइप क्या है. हेलो. So we check for the immunofluorescence. इम्यूनोफ्लोरेसेंस से हमें पता चलता है कि in that stage of the parasite, is there any difference? For example, if the gene is very essential for the parasite survival, we will not get any knockout population. We will not able to make any knockout population, because the parasite will not survive in blood. So we can conclude that the gene is essential in blood stage. But how do we know its role beyond blood stage? Then what we do, we make conditional system. So what are conditional systems in knockouts? कि जेनेटिक जनरल नॉकआउट में हम एक जीन को पूरा डिलीट कर दे रहे हैं, बट इन कंडीशनल सिस्टम व्हाट वी डू, दैट जीन विल रिमेन थ्रू आउट द स्टेजेस of the plasmodium, but it will be deleted or excised from the genome only when you are giving an inducer. That inducer will activate an enzyme that will excise that particular gene. So that is a conditional system. Conditional system help us study role in different stages, like in mosquito, in liver, because blood में तो अगर वो essential है जीन, तो blood में तो वो parasite survive नहीं कर पाएगा उस जीन के बिना. Then how will we know its role? And if we don't know its role, how will we able to make it a therapeutic target? So therefore, to study it, we make conditional system. So this is all we do in our lab. I am also working currently on a particular gene, which right now I can't disclose for obvious reasons. But we make knockouts, and we do in vivo and in vitro analysis as well. So, connecting bacteria and parasites. So, in summary, what I was trying to tell is that whether be it bacteria or a parasite, a parasite is much more complex than a bacteria, but still the outcome is the same for both. Whenever there is a drug pressure, there will be a regulatory plasticity in both bacteria and plasm
Mhm.
plasmodium. They will try to make some survival strategies for them, and due to which these existing drugs will be of no use. So both rely on repurposing existing molecular machinery. That's why studying antimicrobial resistance is very important, and studying their molecular mechanism of microbes is important for searching targets. So the take home message is the same, that different pathogen has same evolutionary logic. Indiscriminate use of drug repurpose their biology for survival strategy, and therefore we need new and promising foolproof target as a continuous need. That was all from my side. I want to really thank Biopractify team for giving me this opportunity to speak up on my subject. Really thank you, sir, and thank you to the entire team. Thank you for having me here.
Thank you very much, Miss Ayushi. And I think we still have a few minutes left. So, if any participant has any question, they can ask them now. Otherwise, we'll proceed to vote of thanks.
Okay, okay.
Any questions from any participants? You can even use the chat box or unmute yourself. Well, I don't think there are any questions from this participants, and I just, I would like to ask a question. How was
Yes,
your
sir.
experience in general, like moving from IIT Roorkee to CSIR CDRI, both of which are extremely, like, significant institutes in
Yes.
whether we see at the developments or the role they play in the educational academia, like sort of environment. So, what was your experience like?
So I had a really good experience at IIT. So, sharing a little, my bachelor's was from my hometown itself, from Gorakhpur. It was from a private college, where we do like BSc in life science in botany, zoology, and chemistry core subjects. So there was no microbiology, no biotechnology. So I got all the exposure. Like I haven't even seen a laminar before my master's, not even a pipette, nothing. So all that I have learned in those 2 years of my masters. So there was a great exposure. What I feel and what I have heard from people during master in India, I don't think any institute other than IIT would give you that level of exposure during your masters. because I was a part of a very good paper. It was in ACS, and I got that opportunity in my dissertation itself. I was working independently, so that was a very great exposure. The mentors were really great. The professors, they believed in you. And I switched to CSIR is because masters was of 2 years, but PhD is a very big commitment, and I wanted it to commit to a specific research lab on only a research environment. So I felt that CSIR would be the best for it, because here I feel that the fundings, the research fundings, are much more better. So there is a much more research work here, much more better. Life is better at IIT. I wouldn't deny, but the research life I feel is better here. So that both institute gave me good experience. So it was nice.
Well, that's really great since you got to, like experience both the institutes
Yeah.
with your master's and PhD.
Yes.
So it is really great, and it is still a dream for a lot of students to,
Yes.
get into any of these institutes as well. So
Yes.
with this, I'll just give a final call for questions. Otherwise, if there are no more questions, then we'll move to the final vote of things. So, anyone, it's your like final opportunity. Okay, then. So, on behalf of Biopractify and the Beyond Research, where curiosity meets conversation team, I extend our heartfelt gratitude towards Miss Ayushi Singh for delivering such an insightful and engaging session. Your journey from bacterial systems to complex parasitic models and the way you connected molecular mechanisms of resistance with the real world therapeutic discovery was truly inspiring and intellectually enriching for our audience. We sincerely thank you for taking time to share your research experiences and valuable perspectives with our participants. Your clarity and depth and passion for infectious diseases and research have undoubtedly motivated many young researchers and students to think beyond conventional path. And the reason that we kept this session during this time is because there are also students who are learning about microbial genomics from one of our programs. They are also participating, so to give them a perspective as in how the field of microbiology and your therapeutics like anti microbial resistance, and this has opportunities and where you can do like something related to it in research. So this was also a part of the reason, and we would like to also thank all the participants for their active presence and thoughtful questions and enthusiasm, which made the session really interactive and meaningful. And lastly, we appreciate the efforts of the collaborators like the ACS chapter who are making this initiative possible. And thank you once again, Miss Ayushi. And we really look forward to more such sessions of you in future. And it was really insightful and interactive and also inspiring for us.
Thank you so much. It's a really great initiative, and I'm glad to be a part of it. And thank you so much.